Our next speaker is Dr. Spencer Diamond, who is a principal investigator at the Innovative Genomics Institute at UC Berkeley, where he leads research on in vitro microbiome modeling. His work focuses on developing systems-level optimization strategies for microorganisms used in industrial, environmental, and biochemical settings. The floor is yours. Thank you.
Good afternoon, everyone, and thank you for inviting me. Thank you to the organizers. What my lab, as they said, focus on is, is really modeling microbiomes as a whole. And I think at IGI, one thing that we're thinking about very broadly is not just the rumen microbiome, but how do we use microbiome modeling and genetic editing to shift the behavior of microbiomes as a whole. And so we believe that the rumen is actually probably one of the best model systems to start doing this type of work in. And so I want you guys to feel empowered in that the rumen is really the place where this kind of basic microbiology and next stage of microbiology where we're going to shift microbiome collective behavior can begin, and that it's really going to be the ideal system to do that. So two core technologies that were developed at the IGI are genome-resolved metagenomics, which was developed by my mentor, Jillian Banfield, and CRISPR-Cas targeted engineering, which I imagine you all are familiar with Dr. Jennifer Doudna. So genome-resolved metagenomics, for those who aren't familiar, is basically the ability to sequence an entire microbiome, recover all of the genomes, so all of the genomic information of all the different bacteria, archaea, viruses, plasmids, everything that's there. So to create a roadmap for kind of what you want to edit. And then the CRISPR targeted editing, this is like your scalpel. This is basically how you're coming in, and you can then modify different pieces of DNA however you see fit. So my colleagues and I in 2022, we put these two technologies together to create a generalized strategy for targeted editing of microbial communities in situ, right? And so even though we didn't do this in a rumen or in a human yet, we were doing it in complex microbiomes that indicated that it was possible to make genetic changes in individual organisms, targeted genetic changes that we did not have to make in those organisms before combining them together in a community. We could do that in the community itself. So, So as we all know, that most of the global methane sources are microbial. So beyond just methane being produced from enteric fermentation, there's methane being produced from waste lagoons, from wastewater treatment, from landfills, many different sources, but it's all microbial. And this is all microbial communities that their behavior together is influencing this methane production. So even though the archaea are the ones responsible, there's many other organisms in the system that are influencing them. And so of course, one of the biggest pieces of this pie is enteric fermentation, which is this miraculous system, but it has a good side and a bad side. And so as a microbial community, it's doing one thing that's great. It's producing a huge quantity of the animal's energy requirements, but it's also potentially releasing a lot of that energy as methane. And so when we think about microbial communities, we think about this very interconnected structure. You have, kind of different trophic levels, just like you're in a forest or a rainforest, right? And you have some animals eating other animals, some animals eating plants, primary producers, et cetera. We have fiber coming into the animal and being degraded through various stages by different microorganisms and ultimately producing things like hydrogen and CO2, which can be used by methanogens. So at IGI, we have kind of a multi-tiered strategy for how we want to modify microbiome behavior. And you could replace this cow with a human with a wastewater treatment plant with whatever you want. But ultimately, we're hoping to start here because we think this is a very tractable problem. So the first step is modeling microbiomes, and that's really what my lab does. And we think about what is each individual species in the system, and ultimately can we identify groups of species that may be competing for the same resources or dependent on each other that might influence the amount of methane or volatile fatty acids that are being produced. Subsequently, we're isolating and also validating these strategies that we would maybe modify some of these organisms using genetic editing. So if we modified, let's say, the purple organism there to be more aggressive at collecting hydrogen and tried to remove some of the methanogens from the system, could we actually shift the equilibrium of the system more towards VFA production versus methane production. We also have teams developing specific genetic tools. So let's say we don't actually currently have the genetic tools to modify this purple organism or hydrogen-utilizing organisms. We are working on developing them directly, and I can talk more about that if anyone's interested. And finally, we have teams working on formulation. So if we have these editing systems, could we deliver them directly to an animal, to a human, and could they then get to the place they need to go and make the edits they need to make? And instead of using things like antibiotics or feed additives, this targeted editing approach is rationally designed, highly specific, and theoretically predictable, and hopefully permanent. So the future directions I'd like to think about with respect to this, especially in the farming community, is that we are thinking about doing this in the rumen of cattle, but things come out of cattle and they end up in a big lagoon like this, and they're producing other greenhouse gases like nitrous oxides, which we haven't heard too much about today, but ultimately are very important on-farm greenhouse gas production. And then potentially this manure is going into a biogas production plant. All of these are microbially mediated processes that are producing these gases. So all of these systems' outputs are impacted by their preceding microbiomes. And ultimately, all these systems really share a lot of similar microbiome structure. And so really understanding and altering any of these microbiome systems, maybe starting with the rumen as we're planning to, has the potential to significantly increase the efficiency of on-farm systems. And so with that, I just want to acknowledge all of the folks who have been involved in this project in my lab, and I'm happy to take any questions as part of the panel.